You know those crazy sci-fi headlines about young blood transfusions revitalizing old brains?
Oh, right. The ones that sound like they're straight out of a vampire movie.
Exactly. It sounds totally absurd. But, behind all that hype is actually a much deeper, very real question for you listening right now. What is your normal blood doing to your brain, like, at this exact moment?
Right, because we traditionally view the blood-brain barrier as this impenetrable fortress. We kind of just think of blood as a delivery service for oxygen and nutrients.
Just a highway, basically.
Yeah, just a highway.
Yeah.
But the reality is that your blood and your brain are actually having this highly complex, continuous conversation. And today we're looking at this incredible new paper from Nature Communications that shows your immune system isn't just fighting off cold, it might literally be dictating how social you feel today.
Which is just a massive paradigm shift, and that is our mission for this deep dive. We are going to trace this conversation from, a single immune cell in your blood across that brain barrier, right down to the specific neurons that control your desire to actually talk to people.
And we're going to unpack every detail of how this works. It's dense biology, but the mechanisms are fascinating.
Yeah, no textbook jargon, I promise. We'll use some good analogies. So let's just jump right in. Where did these researchers even begin to unravel something this complex?
Well, they started with a very specific genetically modified subject. It's called the RAG1 knockout mouse. You'll sometimes hear it called RAG1 null.
Okay, RAG1 knockout. What's special about them?
The critical thing you need to know is that these mice are severely immunodeficient. Because they're missing this one specific gene, the RAG1 gene, they completely failed to produce mature T cells and B cells.
So wait, their adaptive immune system?
Is essentially non-existent.
Right.
It's a blank slate.
Wow. Okay, so why use them?
Because the researchers had this really fundamental question. If an animal has, no T cells, does that actually change its everyday behavior? Specifically, its social life.
Okay, I have to ask, how do you even measure the social life of a mouse? I mean, it's not like you can check their calendar to see if they're going to parties or hanging out.
Right. You use a behavioral assay. It's called the three-chamber social interaction test. It's actually quite elegant. Picture a rectangular box divided into 3 equal rooms with little doors connecting them.
Okay, got the visual.
So in the first phase, which measures general sociability, the test mouse starts in the middle room. In the room to the left, there's a stranger mouse safely sitting inside a little wire cup.
Just hanging out in a cup.
Exactly. And in the room to the right, there's an inanimate object, usually like a little plastic toy, also in an identical wire cup.
So the test mouse basically has to choose who to hang out with.
Precisely. Now, a normal, healthy, wild-type mouse is naturally curious, right? It's social. If you track its movement, it's going to spend significantly more time investigating and sniffing the wire cup with the stranger mouse than it will the plastic toy.
Yeah, that makes sense. They want to interact with a living thing. And what's the second phase?
The second phase measures social novelty preference. So they take that plastic toy out and they replace it with a brand new stranger mouse.
Oh, I see.
Yeah, so now the test mouse has to choose between the familiar mouse it just met and a completely novel stranger. Normal mice will prefer the new guy. It requires, social memory and curiosity.
All right, so we take our immunodeficient RAG1 mice, the ones with 0 T cells, and put them in this 3-chamber box. What actually happens?
They completely fail both phases.
Really, both?
Both. They showed absolutely no preference for the stranger mouse over the plastic toy, and they showed no preference for the new stranger over the familiar one. They were deeply, deeply antisocial. But here is where the researchers found their first massive aha moment. They performed what's called an adoptive transfer.
Which is what, exactly?
They took blood serum, or just purified T cells, from normal wild-type mice and injected them into these antisocial RAG1 mice.
And that actually changed their behavior.
Completely restored their sociability.
Yeah.
Suddenly, they wanted to hang out with the stranger mouse over the toy again.
Wait, hold on. When I think of the immune system affecting the brain, my mind immediately goes to inflammation. Cytokines. Like when you get the flu, your blood is just flooded with inflammatory cytokines, and that makes you feel awful, and you just want to hide under the covers.
Right, the sickness behavior.
Yeah. So if you're injecting blood serum, aren't you basically just washing their brains in cytokines?
See, that is the exact logical assumption, and it's literally the very first thing the scientists checked. But when they analyzed that wild-type serum they were transferring, the circulating cytokines, things like interferon gamma and IL-17, which we know can alter behavior, they were actually below detection limits.
Wait, really? Below detection?
Yes. There was no inflammatory storm happening at all. However, when they checked the blood of the RAG1 mice after the injection, they found specific CD3 plus markers.
Meaning what?
Meaning T cells were now present and circulating. So, cytokines absolutely weren't driving this social rescue. Something else from the normal blood, specifically tied to those T cells, was carrying a pro-social message to the brain.
Okay, let's do a really quick 5-minute recap just to make sure this foundation is solid for everyone listening. Bullet point 1, we have a RAG1 mouse with no T cells, and it has severe social deficits.
Yep, spot on.
Bullet point 2, when we inject normal wild-type blood serum, sociability is fixed. And bullet point 3, we know for a fact it is not a generic wash of inflammatory cytokines causing this.
Exactly right.
So ask yourself, if you're listening to this, have you ever considered that your immune cells might be actively dictating how social you feel today? Because clearly the message is traveling another way, which brings us to the actual messengers.
Right, the extracellular vesicles, or EVs.
EVs. I have this concept. I like to picture these as microscopic biological mail trucks.
That's honestly a great analogy. If you imagine your body's cells as houses, EVs are these microscopic, lipid-wrapped mail packages sent out into the bloodstream.
Just floating in the blood.
Yeah, they're incredibly small membrane bubbles, and they carry really diverse cargogenetic material, proteins, lipids, from one cell to another.
But if they're just like invisible bubbles floating in an ocean of blood, how on earth do researchers isolate them to actually study them?
It is an incredibly intense process. To pull these tiny packages out, they use a technique called differential ultracentrifugation.
Which means spinning them really fast.
Extremely fast. They take the blood sample and spin it in a centrifuge at 100,000 times the force of gravity.
100,000 Gs. That is an insane spin cycle.
It has to be, just to force those tiny, almost weightless particles down to the bottom of the tube. And after that, they use size exclusion chromatography, kind of like a molecular coin sorter, to filter them by exact size.
And then they just look at them.
Yeah, they literally count the individual nanoparticles using lasers with nanocyte tracking and take pictures of them using an electron microscope.
Wow.
And when they did all this, they found that our antisocial RAG1 mice did have EVs in their blood, but they were missing a very specific subset, the ones produced by T cells.
Okay, so the million-dollar question. What happens if we take those purified EV mail trucks from normal wild-type blood and inject just those packages directly into the antisocial mice?
Sociability is completely restored. Just the vesicles alone were enough to fix the behavior.
That is wild.
And it gets better. They didn't stop there. They tested this on 2 other completely different, well-known genetic mouse models of autism, the SETNAP2 knockout and the SHANK3 knockout models.
And both of those naturally have social deficits.
Exactly. And injecting the normal blood EVs improves sociability in both of those models, too.
Now, I have to ask the obvious follow-up. If I take these normal pro-social blood packages and inject them into a completely normal, already social mouse...
Does it become an ultra-social party animal?
Yeah. Did they just start throwing a frat party in the cage?
No, and that's a really crucial distinction to make. When they gave WTEVs to normal mice, their sociability didn't increase at all. They didn't change.
Oh, interesting.
Yeah, these packages don't create a superpower. They restore a deficit. They return a broken system back to baseline.
Okay, so we know these mail trucks are in the blood, and we know they somehow fix social behavior, but the brain is protected by the blood-brain barrier. It's a literal fortress. How do we know these tiny bubbles are actually getting inside the brain?
The scientists used 2 really brilliant visual labeling methods to prove it. First, a chemical method. They took the normal EVs and dyed them with a special red lipophilic dye called PKH26.
So now we basically have millions of glowing red mail trucks in the blood.
Exactly. And the second method was genetic. They collected EDs from a special line of mice called MTMG mice.
What's special about them?
Their cell membranes naturally fluoresce bright red because of a TD tomato protein. And since EVs are made of cell membranes, every single package these mice produce naturally glows red, no chemical dye needed.
That is so incredibly clever. So they inject these red glowing packages into the blood. What do they see?
Within just 1 hour, they found those glowing red dots deep inside the brain parenchyma. They breached the fortress.
In 1 hour, where did they go?
Specifically, they clustered in the medial prefrontal cortex, the hippocampus, and the cerebellum. But here's the most fascinating part. They didn't just wash over the whole brain randomly.
Right, they actually had specific delivery addresses.
Yes. When they tracked them, they saw the EVs selectively entering 2 very specific types of cells. The neurons, those are the NSE plus cells, and the microglia, the IVA1 plus cells, which act as the brain's local immune cleanup crew.
Did they hit anything else?
No, they completely ignored the other support cells like astrocytes and oligodendrocytes.
So to summarize the second major piece, and I want you listening to really think about this, if microscopic bubbles from your blood are slipping into your brain's command center right now, what specific messages are they programmed to deliver? Because they are acting like highly targeted smart mail.
Exactly. They cross straight from the blood, seek out neurons in key areas like the medial prefrontal cortex, and dock.
Okay, so let's zoom in on the medial prefrontal cortex, the MPFC. That's essentially the brain's command center for social behavior, right?
Yeah.
It regulates how we interact. What was actually going wrong in that specific area for our antisocial mice?
In the RAG1 mice, the MPFC is deeply troubled. It's what neurologists call hyperexcitable. There is simply too much chaotic electrical noise.
How do you even measure chaotic electrical noise in a living brain?
They look for a specific protein called CFUS. You can think of CFUS like a tiny light bulb that turns on inside a neuron only when that neuron fires an electrical signal.
Right.
And when they looked at the prefrontal cortex of the antisocial mice, it was lit up like a Christmas tree. Specifically, the CAMKI alpha plus excitatory neurons were firing way, way too much. The brain was just overwhelmed with static.
But, correlation isn't causation. How did they prove that this specific static was causing the antisocial behavior?
They used this fascinating, highly technical method called chemogenetics. Specifically, a tool known as DREADDs.
DREADDs. What does that stand for?
It stands for designer receptors exclusively activated by designer drugs.
That is a mouthful.
It really is. But the concept is basically installing a biological remote control in the brain.
Okay, walk me through how you install a remote control in a living brain.
The scientists use an AAV, an adeno-associated virus. It's a harmless virus that acts like a microscopic delivery vehicle. They program it to install a custom artificial receptor switch called HM4DG directly onto those loud hyperactive neurons in the MPFC.
Okay, but this new switch doesn't do anything on its own.
Right. It's totally invisible to the body. It just sits there doing nothing.
Until you use the remote.
Exactly. The scientists then inject a custom drug called CNO into the blood. The CNO is the remote. It circulates, finds only those custom switches, and activates them. And when activated, the switches force those hyperactive neurons to calm down.
They literally just hit a mute button on the prefrontal cortex.
Yes. They turned down the volume.
And what happened to the mice when they turned the volume down?
It was immediate. When they artificially hit that calm down button with DREAD, the RAG1 mice suddenly became social again.
Just from quieting the noise.
Just from quieting the electrical noise. The desire to interact completely returned.
But wait, mice in the real world don't have scientists with viral remote controls fixing their brains. How does this connect back to the EV mail trucks we were talking about?
Because the EVs do exactly what the remote control did, but naturally. Even without the high-tech switch, injecting wild-type EVs naturally reduces the CFOS hyperexcitability.
So the EVs are the natural volume knob.
Exactly.
That is incredible. But how do they actually turn down the volume biologically?
To figure that out, the researchers used patch clamp brain slice electrophysiology. They measured the spontaneous inhibitory postsynaptic currents, or SIPSCs.
Okay, inhibitory meaning the signals that tell the brain to stop firing.
Right. If excitatory neurons are the gas pedal of the brain, inhibitory currents are the brake pedal. They keep the system from spinning out of control.
And let me guess, the antisocial mice had bad brakes.
Terrible brakes. The SIPSCs were dangerously low. But when they applied the normal wild-type EVs to the brain tissue, the brakes were fully restored. The system calmed down.
Okay, let's do our 10-minute recap. Bullet points. 1, WTEVs travel to the brain. 2, they target specific neurons. 3, they calm down the overactive MPFC. And 4, they do this by restoring the brain's inhibitory brakes, those SIPSCs.
That's the exact pathway.
So when you, the listener, feel totally overwhelmed and just want to withdraw in a crowded room, could it be a temporary lack of these microscopic breaks in your own prefrontal cortex? Because it leaves the biggest mystery completely unsolved. What is actually inside those EV mail trucks that has the power to rebuild the brain's brakes?
Right, we have to unpack the cargo. What is the actual biological mail?
The paper says the cargo is microRNAs. What exactly are those?
MicroRNAs or miRNAs are these tiny genetic sequences. Now, unlike messenger RNA, they don't build proteins. Their entire job is to be a genetic silencer.
A silencer?
Yeah, they seek out specific messenger RNAs and destroy them, which prevents certain proteins from ever being built in the first place.
Okay, but before we talk about which proteins genes they silence, I want to go back to the mail truck idea. How did the scientists prove these fragile little strands of RNA were actually safely riding inside the lipid vesicles and not just floating around loosely in the blood?
They ran this incredibly elegant experiment using RNAs and Triton X100.
Okay, what does RNAs do?
RNAs is an enzyme that's basically a molecular shredder. It aggressively destroys any exposed RNA it touches, but when they treated the blood sample with it, the specific microRNAs they were looking for survived perfectly.
Because the lipid envelope of the EV protected them from the shredder.
Precisely. It's armor. And to prove it, they then added Triton X100. That's a harsh detergent.
Oh, I see. Like dish soap breaking down grease. So they literally popped the lipid bubbles.
Yes. They popped the bubbles and then applied the RNase shredder enzyme, and instantly the microRNAs were destroyed.
That is such a cool way to prove it. It proves the cargo is absolutely inside the armored trucks. So they sequence the cargo inside these popped bubbles. What specific genetic codes do they find?
RNA sequencing revealed 2 specific microRNAs that were completely missing in our antisocial RAG1 mice. They are called miR23A3P and miR1033P.
miR23A3P and miR1033P. And what do those specific genetic silencers target?
They target a gene called PKCE, which makes an enzyme called PKCε.
Okay, why do we care about PKCε?
Because when PKCε goes unchecked, its job is to actually remove GABAA receptors from the brain's synapses. And GABAA receptors are the primary brake pads of the brain. They create those inhibitory currents we talked about.
Wait, let me make sure I'm picturing this right. If the GABAA receptors are the brake pads on a car, this PKCε enzyme is basically a rogue mechanic running around stripping the brakes off.
That is a perfect analogy, yes.
And because these immunodeficient mice lack the microRNA to stop him, their brains are just speeding out of control.
Exactly. Because they have no T cells, they aren't sending that specific microRNA to the brain. So the rogue mechanic runs wild, strips the GABAA brakes, and you get that hyper-excitable, static, and antisocial behavior.
It's a total breakdown of the system. But then the normal EVs arrive. Let's map this whole story. The EVs cross the blood-brain barrier, they dock with the neuron, they drop off their cargo, the microRNA, and this microRNA acts like a bounty hunter, hunting down and destroying the blueprints for that rogue mechanic.
Yep. The mechanic is gone, the GABAA brake pads return, the electrical noise drops, and the mouse wants to socialize again.
It is a stunning biological circuit. Did they prove this was the exact mechanism?
They did. They performed one final brilliant proof. They took empty EVs from the antisocial mice and artificially loaded them with synthetic, laboratory-made mimics of just those 2 microRNA.
They packed their own fake mail?
Yes. And injecting those artificially loaded EVs perfectly replicated the natural wild-type rescue. It lowered the rogue PKCε, increased the GABAA brakes, restored the electrical calm, and completely rescued the mouse's sociability.
Okay, final 15-minute recap. The cargo is miR23A3P and miR1033P. They are protected inside the EVs. They stop the PKCε enzyme. And this restores the GABAA receptors, which are the brakes.
That's the whole pathway in a nutshell.
So think about the sheer precision of this for a second. A single tiny RNA molecule altering how you connect with others. What other hidden genetic codes are driving your daily interactions? It's crazy. But that leaves one final piece of the puzzle, the origin. How do we know these specific microRNAs absolutely, without a doubt, come from T cells?
Right, because EVs are produced by almost every cell in the body. So to prove the source, the scientists first took T cells and grew them in a petri dish. They collected the fluid, isolated the EVs that those cultured T cells secreted, and sure enough, they were packed with our target miRNAs.
But, growing cells in a dish is one thing. How do you prove it in a living, breathing animal?
They did a knockout proof. They genetically engineered a completely new mouse that was totally normal, except it lacked the gene for just that one specific microRNA, miR23A. So it's a miR23A knockout mouse.
Okay.
They then purified the T cells from this mouse and transferred them into our antisocial RAG1 mice.
So the antisocial mouse now has a working immune system. It has T cells circulating in its blood, but those T cells are essentially firing blanks. They don't have the specific cargo to put in the mail.
Exactly. The T cells survived and circulated perfectly fine. But because they lacked that one specific genetic cargo, they completely failed to rescue sociability or the electrical brain currents. It proves the T cells absolutely must have that specific miRNA to influence the brain.
Now, we spent this entire time talking about mice, genetic knockouts, brain slices. It's fascinating biology, but what does this mean for us, for human beings listening to this right now?
Well, this raises an incredibly important question about clinical relevance, and the source material actually provides some compelling graded evidence that this isn't just an isolated mouse phenomenon.
Really? Humans too?
Yeah. Clinical studies have shown that patients diagnosed with autism spectrum disorder, as well as patients with schizophrenia, show significantly altered levels of these exact same microRNAs in both their blood and their postmortem brain tissue.
The exact same genetic silencers.
Yes. For example, in schizophrenia patients, the levels of that specific microRNA are notably decreased in peripheral blood. In ASD patients, it's consistently decreased across multiple independent studies.
Wow.
So the exact same deficiency causing social deficits in the mice is being observed in human conditions characterized by social and cognitive challenges.
Which totally makes you wonder about the future of medicine. Could tracking these immune packages in human blood one day replace, like, subjective psychiatric questionnaires? Imagine going to the doctor because you're suffering from severe social withdrawal, and instead of just filling out a form about your feelings, they literally check your blood to see if your T cells are sending the right mail to your brain.
It represents a massive paradigm shift in how we might diagnose psychiatric conditions. We're moving from just observing symptoms to actually measuring the periphery-to-brain communication network.
Okay, let's synthesize all of this, because the journey we just took is wild. We started with an immune cell living in your blood, which packs a tiny piece of genetic code into a lipid bubble. That microscopic armored truck travels through your bloodstream, breaches the heavily guarded blood-brain barrier, and selectively docks with a neuron in your prefrontal cortex. It releases its cargo, stops a rogue enzyme from destroying your brain's electrical brakes, and suddenly the static clears and you feel the desire to go socialize.
And all of that is happening constantly, perfectly, just below your awareness.
Yeah.
It truly highlights that the brain isn't an isolated supercomputer sitting in a jar. It is deeply, continuously integrated with the immune system. You really cannot fully separate neurology from immunology anymore.
And that leaves us with a final, provocative thought for you to mull over. If our immune system, specifically our T cells, is constantly sending these social regulating packages to our brain, what happens when our immune system is compromised?
That's the big question.
Right. Think about chronic stress, autoimmune flare-ups, or even just weeks of terrible sleep. Could these things be directly rewriting our neurobiology by disrupting this vesicle mail delivery? Could they be the actual biological cause of us wanting to socially withdraw when we're stressed or sick?
It certainly implies that treating social and psychiatric conditions in the future might look very, very different than it does today.
Exactly. Could the future of psychiatry look less like traditional brain chemistry modification with daily pills and more like immune system optimization? Imagine a future where we treat severe social anxiety by drawing your blood, engineering your own natural extracellular vesicles to carry more of these pro-social microRNAs, and infusing your own optimized blood back into you. Autologous therapy. We wouldn't be searching for some mythical fountain of youth. We'd just be fixing the biological mail your body sends every day.
It's a completely new frontier of medicine.
It really is. Well, thank you so much for joining us on this deep dive into the microscopic highways of the body. Keep asking questions, keep looking deeper, and stay insanely curious.